This is episode 119 of the Immunology Podcast, Systemic Lupus Erythematosus with Dr. George Robinson. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. George Robinson from the University College London on the podcast to talk about his research on juvenile-onset systemic lupus erythematosus. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Do you need to isolate human regulatory T cells? STEMCELL Technologies have created a step-by-step video showing you how to isolate human regulatory T cells from large volume samples using EC-CEP technology. The video goes over each step in detail so you can follow along and obtain the cells you need for your downstream experiment or analysis. To watch the video, go to stemcell.com/Treg isolation video. So, do I need to say happy Thanksgiving? Are you getting some turkey anytime soon, or?
It's like 2 weeks away.
Oh,
Yeah.
still preparing.
Yeah. It's
Okay.
very late this year. Usually it'd be next week, but because of like the X Thursday of the month is just off, it's as late as it possibly can be. Getting ready. No turducken this year. We did that once, though. Do you know what a turducken is?
A turducken?
Yes.
No. Enlighten me.
You take, you debone a chicken and put stuffing inside of it and stuff that in a deboned duck with stuffing inside of it. And stuff that into a bone or deboned turkey. And then you cook the whole thing.
Oh my God.
Sometimes with Cajun spices, you have different flavorings for each layer. Or, you could deep fry it too, but usually you do that separately. But yeah, turducken is a...
Sorry, do you deep fry the whole thing?
You could. You can deep fry a turkey or a turducken.
The thought of frying all of that, I think my liver
you do it. It's no different than frying a turkey, right? You got to put it on a rack and drop it in. You can't do it frozen, otherwise it explodes. Every year, lots of Americans end up in the hospital from deep frying, something that's frozen, and so the water like immediately vaporizes and carries the boiling oil onto people. You can't do that. But yeah, turducken is a time-honored American tradition I've done once.
That is such an American thing.
It is.
I cannot imagine any European thinking that's a good idea.
Well, you guys stuff pastries with cream and stuff. We stuff meat inside the meat.
Inside the meat. It's 3 layers.
Stuffing meat, stuffing meat.
Different
6
meat.
layers.
3 different birds. One inside of, oh my God.
With 3 stuffing.
All right.
In layers.
With 3 stuffing. No.
Yeah, it's glorious.
Why do you have to overdo? Oh gosh. Well, if that makes you happy, America. I know that you have a lot to cope with, lately.
Hey, our government's open today. They signed the bill. Our government opened back up.
Congratulations. I mean, let's not go over why it was shut down in the first place, but good for you.
We'll have our flights back in a week or so. It'll be wonderful.
Yeah. Seriously, that's one thing I was really starting to get concerned about after.
Yeah.
Then now we can fly to go eat your turducken at your family home.
You know what I'm going to talk about today, Brenda?
What are you good turduckens? I hope not.
Something that has a longer half-life than the American government shutdown, which are long-living plasma cells and IgE producing plasma cells.
Excellent. Bring it. I'm all ears.
Yeah, all right. I figured that segue would be a little unique. Why not?
Amazing.
All right. So this is in Immunity coming out November 11th. So in the future, gotta love it. Paper name is Long-Lived IgE Plasma Cells Persist in Secondary Lymphoid Tissues using a Navito-Clax-Sensitive Survival Program. First author is Zhaoji Ding. Last author is Marcus Robinson. So, question is, how long do IgE plasma cells live? Right? And they answer that in this paper. Thank you, I'm now done. No. So, that is the moral of the story. What they do is they look at, they use a tracing system that's Cree-induced that puts a human CD4 in front of a survival gene, MCL1, after Cree, and use that as a lineage tracer for timing. And so they use a dust mite system in mice, infect all of the mice with, expose these mice to dust mites, get IgE, and then they time how long the signal persists. Because what will happen is the recombination will persist, but the fluorescence tag doesn't. And so what's cool is they find there's two populations. There's a short-lived and then a much rarer long-lived. And they find that the duration of short-lived is directly proportional to how long there's exposure. So the more, they live a certain amount of period after exposure. So if you stop dust mites, they peter off. But more exposure doesn't lead to more long-lived at all. They have this whole separate programming. CXCR4 they implicated as being important for accruing in the bone marrow. And then they actually were able to get one more insight from this, which was cool, which is they were able to figure out that they used this MCL1 reporter, and then they figured it was actually tied to what they were studying. And so there's this BCL2, BCLX, BCLW, that set of genes, anti-apoptotic molecules, and those are important for the survival of these cells. And so when you hit them with an inhibitor, that's that Navito-Clax inhibitor, they all pop goes the weasel. So point being, it seems to be that there's a small subset of IgE cells that persist well past producing cells that persist well past the exposure period. Not just the IgE, which we knew IgE can be long-lived, but there's actual plasma cells producing it, which is not known before. There's always a question because there are also these short-living cells. And so they were able to sort it out and then get your answer. So I don't have to describe more than that. Basically, that is the paper. They go through the different steps of tracking it and look at some of the tissue programs. But that's really in the end the point, is there is a subpopulation of very long-lived, but it's very bifurcated.
Mm. And where are they found again?
Where are they found? Most of the bone marrow, but also the mesenteric lymph nodes.
Okay.
Spleen.
Yeah. So then it's not only like the typical memory B cells that we think are the long-lived ones, but also these plasma cells that are actively churning out antibodies. They are themselves long-lived. And this is kind of what we didn't necessarily expect.
Yep.
All right. Nice. And do they give rise to other plasma cells, or is that also?
They can. They persist without a lot of self-renewing. The ones that self-renew are the short-living ones, and then they peter out.
Okay.
But there is some proliferation of them. And they look at that using Ki-67.
All right.
But they hang out a really long-ass time. Months.
Nice. Okay. I guess always the idea is that only the memory cells that are not actively making antibodies are the ones that are staying. And but I guess it also makes sense to
we
have
know there's active IgG plasma cells that hang out.
Yeah.
That's how else would you have sterilizing immunity for anything, right?
Yeah.
But IgE cells were always like, well, there's some data that's short, some that it's long. Guess what? It's both. It's a bimodal population that they mathematically model their data and show that it only works when you have two different half-lives.
Mm-hmm. Oh, okay. There's even a mathematical model where look at that.
Two populations. Look, when you model the data, it requires two different half-lives to model. Like, yeah, because there's two populations. Thank you. It was tautological, but necessary.
All right. Okay. Well, moving on, but in a, I think, kind of similar topic, I'm going to talk about a paper about peanut allergies. Do you know anybody with peanut allergies, Jason?
A few people. Mostly it just affects what my kid can take to school, which is annoying.
I don't hear a lot about people with peanut allergy here. I don't think I've ever met anybody that knows anybody that has a peanut allergy, to be honest. That's very strange. But yeah. In any case, peanut allergy, not great. But there are some treatments to it, and oral immunotherapy is one of the potential ways to reduce the peanut allergy and try to make it less, I guess, life-threatening or less disruptive to normal eating. So this paper is called Peanut Allergy Oral Immunotherapy Drives cell multi-omic changes in peanut reactive T cells associated with sustained and responsiveness, published in Nature Immunology, November 4th. So, as I mentioned, oral immunotherapy is basically now, I think, is the only approach that you can try in order to reduce the allergy to peanuts. And people, for such a treatment, people basically um, treated with incrementing amounts of the. They start with very small amounts of peanut antigens, peanut proteins, and they increase. And basically, in this way, you can reduce the chance that people will actually go into an anaphylactic shock upon eating peanuts. And so in this paper, what they did is they're following a clinical trial in which they uh, treat patients with oral immunotherapy, and they try to understand. They look into the T cell compartment and try to understand what's happening with the T cells in this patient. So, um, as part of this a clinical trial, they have two different treatment conditions. So they have in all cases they have some placebo controls, but they also have some people in which they are um, treating people with increased amounts of peanut. I have to say, actually, I don't know. Do they treat them with like peanut protein? What exactly are they using? It's not clear to me. What I'm looking at and I just, yeah, peanut. They're treating them with peanuts, but what exactly is it that they are injecting to this patient or treating this patient? No, or immunotherapy? No, um, peanut protein. So they have increased amounts of peanut protein, and then they sustain a high dose around 4,000 milligrams or 4 grams of peanut protein for many, many weeks. This is a 2-year treatment. So they spend a whole year increasing the dose, and then a whole year treating these people with 4 grams of peanut. And then they have another few months in which they have two groups, either people that go into avoidance, so they stop taking the peanut protein, and people that have a maintenance low dose of 0.3 grams of peanut protein. And that is kind of the different groups that they have and they're analyzing. And what they see is that some people will um, be desensitized. So some people will become desensitized at the end of these 2 years of treatment. And some people will sustain this desensitization after not having been exposed to the peanut protein for a few months. But not everyone. And it's actually, the numbers are very sad. So I think about half of the participants actually achieve some kind of desensitization. So, and some what is called sustained and responsiveness. About half of the participants do this. What they see is, so they do a lot of analyses on, they stimulate the T cells from the patients, so from blood of the patients with peanut protein solution, and they look into different T cell populations based on activation markers, CD137, or also known as 4-1BB, or CD154, also known as CD40 ligand. And these are usually used to recognize self-reactive or reactive T cells, conventional T cells, and also regulatory T cells upregulate CD137. So they also use that to kind of differentiate conventional from regulatory T cells. And what's interesting is that they see that in those patients that kind of the most interesting result of this paper is that those patients that um, achieve a sustained and responsiveness. So after months of not being exposed to peanut protein, they still don't develop a reaction to it. They seem to shift. They show a shift of a mostly Th2-like response towards a more Th1 response. And they see, or also patients that respond better, they usually have a baseline, they have differences in the baseline. But what they see is basically a loss of Th2-like cells, either specific clones. They see that they lose these clones, or they see some clones that become more Th1-like, and that seems to correlate the best with loss of cell of kind of improvement of the allergy and and desensitization, more and sustained disease and responsiveness in these patients. So, because the question always is, what's happening? Is the Th2, you have a population of Th2 T cells that are, mediating this response, that are making antibodies, are sorry, and also that activate, basophils and activate these uh, allergic response. So is it happening? What happens if you're constantly stimulating the cells? Is it that the cells, they go away, they become allergic, or do they kind of mutate into something different that does not activate basophils? And that seems to be the case. Combination of all, because they see some clones that seem to disappear. But it's really hard because there's a lot of clonal diversity, so they have a hard time finding enough kind of clonally expanded reactive T cells. But I think in general, the most convincing part is this shift away from Th2 into Th1, associating with kind of best response to treatment in a way.
So I have to be cautious on this because
Mm-hmm.
you unwittingly stepped into something that so Nestle used to own this drug. This is called Palforzia. It's
Mm-hmm.
FDA-approved peanut protein for peanut allergy, and Nestle's pharma division is called Aimmune, and Aimmune created Palforzia and then sold it off, and then Nestle sold it off after buying Aimmune because of slow sales. Like fundamentally, like you have to
Mm.
have this observed while administering it, and there's a lot of barriers to its use and
Yeah.
practicality. It's been really tough to be even though it's a wonderful drug from a certain perspective, you can also just eat peanut butter in its very small amounts, right?
Yeah. Yeah.
Or a lot of the label around it made it very hard to do practically, and the reimbursement and stuff got tough. And so Nestle ended up selling it off before I joined. But this trial is not new, right? This is a new analysis from old trials.
Mm-hmm.
And this is right up that this is Palforzia, which
Yeah.
is my company's old drug.
Interesting. It's just a small world.
It is.
Oh, sorry. There's one more thing that I also found, which is increased proportion of CD39 positive cells, presumably regulatory T cells. That's another thing that associates with sustained unresponsiveness. So T-Rex coming at it again, you know?
T-Regs are a key thing on this one.
Yeah.
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 All right. Well, then I guess it's my turn to also talk about nothing that has to do with work, which is in nature, lymphoid gene expression supports neuroprotective microglia function. So first author is Pinar Ayata, last author is Ann Schaefer, coming out the 5th of November in Nature. So I wish this paper said more. I'm going to say I don't know why it made Nature on its own. It's cool, but it lasts that second order pop, I think, that I usually expect. There is this transcription factor PU.1, which is downregulated following microglial contact with plaques. That's known. And it's also known that how the microglia behaves with amyloid plaques in Alzheimer's disease is either bad or good. So they establish here essentially that lower PU1 expression in microglia reduces amyloid disease severity in mice. And it is, and so, okay, so that's cool. They find that early, but then they also link it to the fact that when this PU1 drops, you have expression of immunoregulatory lymphoid receptor proteins. And they kind of talk about it in the text, but they basically just say, go see the figure and walk away. So I was kind of annoyed that like, they didn't say, hey, they just like, it's a general pattern. There you go. And they kind of stopped. But they did pick up on CD28 and say that CD28 needs to be there as part of it to protect. If you ablate it, then the effect of the PU low goes away. and then they stop. And this is where I'm like, how is this nature? Right? Because they say, hey, you need this co-stimulatory and co-inhibitory receptor proteins. You need both of them. Because CD28 I don't associate as particularly inhibitory, right? It does lots, but they just say there's this immunoregulatory profile. CD28 is important, and low PU1 plus CD28 is necessary for this microglia, and doing so makes there be less neural inflammation in the presence of plaques and thus less Alzheimer's symptoms. How the PU1 is driven, not there. What CD28 is doing to the cells other than making them have less inflammation, not explained. What the signaling cascade is, not explained. But why is this immunology? Well, again, to the point, these microglia, which are kind of immune cells, right? I mean, they are. They're not classical, right? They're like a tissue resident population that's specialized, like enterocytes are immune cells, or Kupffer cells are immune cells, right, in the liver, right? They're kind of like that, right? They're like a specialized.
But the Kupffer cells are the macrophages of the liver. Of course,
but they're no longer
immune
called
cells.
macrophages, right?
But that's how I learned it.
And microglia are,
the macrophages of the brain. No? Okay.
Right.
Go on.
And enterocytes are the macrophages of the intestine.
Oh, God. Sure.
Right. But they're distinct enough from their original function. They got a new name, right?
Mm-hmm.
But this regulatory program has to pop up for it to function this way.
All right.
So I'm like, this is cool, but it was lacking a lot of that mechanistic punch I was like expecting in the later half of the paper.
Yeah. Yeah.
They just really show that these immune cells interact on the plaques. And there's SKY-PLC gamma 2 signaling, which can be triggered by the knockdown that leads the immunoregulatory type, but there's not a, I just want the thing.
Yeah.
I didn't get the
Jason
thing.
was the thing. He didn't get the thing.
Not there. That's all I got.
Yeah.
But I was hoping for like that punch of like, oh, look, CD28's really doing this here. And it's a co-stimulatory receptor, but it's causing decreased inflammation. That's cool. Yeah. Tell me more, please.
Yeah. Yeah. So, not to, I think, it is not a detriment of the authors, but it's actually like, we want more. This is very interesting. We want more.
Yeah. No, it's a really good paper. My point is like, sometimes like, oh, this is nature, and nature's usually like, where's my 10 extra mechanism steps?
Yeah. Yeah. How long did it take for the revision?
Let's see.
Is that there must be somewhere to be found about his article. Well, just a year of revision. Pretty good.
Yeah.
Pretty standard. Just a year and a month. Okay, so before I forget, I realized that I did not talking about authors. I did not give credit to our previous the peanut allergy story. So in Nature Immunology, and first author is Xiaori Han from last corresponding author Carrie Nadeau at Harvard Medical School. Okay, so for my second story of today, also talking about unwanted immune responses, or about kind of. This is a cool paper from corresponding author Alessandro Sette from La Jolla Institute for Immunology. We've had him in the show previously. First author Tanel Michaelis. And the article is called Autoimmune Response to C9orf72 Protein in Amyotrophic Lateral Sclerosis, aka ALS. And I like this paper because I should have maybe a special segment in this podcast called Everything is Immunology. Yay! So in this paper, it's one of the first, I think, according to the authors, and I could not find evidence against it. It's the first evidence that there is an antigen-specific immune response involved in ALS. And it's not clear what really the implications to disease are, and I'll get to that in a second. But it's interesting because they do find antigen-specific T cells that are relevant to the system at hand. So ALS, we know, neurodegenerative disease, really, very tricky disease, very short life expectancy, and you have this extensive damage to oligodendrocytes. You have loss of motor neurons, paralysis, and I don't know a lot about the disease, but it's known that there's neuroinflammation is seems apparent in the tissues, and there is high levels of T cell infiltration that have been observed in this disease. Um, However, the main would say that the most of the source of ALS, so associated with the disease, is some genetic mutations, and some of the most common, about 40%, include mutations in a protein known as C9 or 72, and these mutations generate is a hexanucleotide repeat. The way that this affects the neural function is through impairing autophagy and lysosomal homeostasis and general kind of this toxicity that damages the neurons. And what they look at in this paper is that they show first that if they take blood from the participants with individuals with ALS, and their counterpart healthy controls, they generate peptide libraries for different proteins that are associated with ALS, amongst them C9 or 72. And they show that patients with ALS have increased amount of T cells that are responding specifically to this peptide, so peptides from this protein, and not the other proteins, and not, for example, controls with EVV proteins. So there's no difference between controls and ALS patients. And they really characterize this recognition. They show that there's certain peptides that are more likely to be recognized. And when they look into the phenotype of the cells that are C9 or 72 specific, what is interesting is that they see that among the things that they produce is IL-10 and IL-5, which are more, I would say, a bit more immunomodulatory, not necessarily inflammatory. And actually, the production of anti-inflammatory IL-10 seems to be associated with those patients that have a longer predictor survival time. And they suggest that maybe these responses are not detrimental, but might actually be providing some kind of anti-inflammatory response. The question, of course, is what are these T cell responders doing? Are they causing the disease in any way? Doesn't seem to be the case, but they might become T cells might become activated, because if you're having this protein that affects autophagy, you might get an increased presentation of this protein by immune cells that are picking up, macrophages that are picking up these dying cells, and they might be cross-presenting these antigens, these internal antigens, to the T cells, and that's what might be driving the increase in the T cells recognizing this particular protein. So what I think is interesting is that the fact that it's not, you see T cell responses, but they seem to be just accompanying the disease, and it's not clear whether they are making it worse or not, or even helping, if they are, in fact, the IL-10 that they're making has some kind of anti-inflammatory function. But this is the first time that they show that one of these proteins that are directly related to ALS also seems to mediate a T cell response that adds to the milieu of cytokines and effectors that are going on in the brains of or in the neural system of patients with ALS.
It's interesting. ALS has been one of those things that's really hard, right? So if it's really an immune condition, that's fascinating.
Yeah. Yeah. So that's, I don't know a lot about ALS. I just read a bit, preparing for this podcast. I'm like, oh man, this is really complicated. It's like people don't know a lot about this. And the involvement of the immune system. And yeah, I thought it was really cool. I'm working a lot on understanding antigen specificity in autoimmune diseases. So for me, this is like, oh, this is very interesting, but also interesting because it doesn't seem to, the T cells are there, but it's not clear what the consequences are of their presence.
Yeah, no, that's another one I'd love to see fixed.
Yeah.
There's been all types of stuff and it just hasn't
Yeah.
worked.
Yeah.
All right. Well, we're going to talk about another sticky disease here with Dr. George Robinson at University College London in a moment. But before we get to that, working with regulatory T cells? Get a free wall chart summarizing the development, phenotypes, and functions of regulatory T cells. To learn more, visit stemcell.com/T-cell-wall chart.
Now, for the second part of our show, as always, we have a great guest joining us today. We have Dr. George Robinson. He is Principal Research Fellow at the University College London. And his research is aimed at understanding autoimmune disorders such as SLE, systemic lupus erythematosus, a really hard word for non-English speakers, I would say. And I think has really cool also research on the effect of gender on these autoimmune disorders. So, Dr. Robinson, thank you for joining the Immunology Podcast today.
Thank you for having me. Nice to meet you, Brenda and Jason. Looking forward to our chat.
All right. I think it's really cool because I think after being made world famous by, I would say, House MD lupus, it's like this. Sometimes I feel like it's a kind of an umbrella term, or like it's many diseases in one in a way. Would you like to maybe share with our listeners when we talk about SLE? What are we talking about, and what is the current status of this disease and the clinical side of it?
Yeah, it's a really good question. It's a very complex disease, and importantly, it's really heterogeneous across patients, which makes it very interesting to study, but also makes it very difficult to study because every patient presents differently with their symptoms or how the pathogenesis develops, and their immunology isn't necessarily the same. And that can be due to different triggers, different causes, different genetic backgrounds. So we have monogenic and polygenic lupus. So typically in younger patients, you'll have a monogenic phenotype occasionally, which happens in a small percentage of patients, but essentially they develop the disease due to a genetic mutation. However, when patients develop it more closer to adulthood, that can be due to cumulative stress or other triggers on the immune system, such as UV exposure. As I'll talk about later, being female is a big cause of lupus or a risk factor, and that makes it difficult to diagnose. So until recently, the diagnosis was sort of an average of around 7 years for patients to get a diagnosis across the world, which just highlights why it's so difficult to diagnose. And it's due to that overlap with different diseases, as you mentioned. So a lot of lupus patients can get overlapping symptoms of rheumatoid arthritis, but loads of other autoimmune diseases that overlap. And typically lupus patients don't necessarily just have lupus. They can have other aspects of autoimmunity as well. I'm sure we'll come on to it, but treatments are certainly improving, and there's a lot of clinical trials currently running. And that's not just beneficial for lupus patients because of this anti-inflammatory mission, essentially, to get that inflammation down in this chronic inflammatory disease. It can have a beneficial effect for lots of other diseases as well. So the development of lupus treatments, and likewise, there's a lot of treatments that are being repurposed from, for example, cancer therapy in the immunology world, such as CAR T-cell therapy, which is a major breakthrough in lupus currently, which is having a massive beneficial effect on certain patients. So yeah, it's interesting to study, but it's also very tricky.
I guess just to set the record straight, what are the target, or how does a person get diagnosed with lupus? What is being recognized? What antibodies are we seeing? What is the diagnosis based on?
Yeah, so diagnosis is based on, they will do blood tests, but also a lot of other testing. But the blood tests are typically looking for anti-double-stranded DNA antibodies or anti-nuclear antibodies. So these are one of the primary causes of lupus. So what happens is that patients will have an excess of these anti-double-stranded DNA in their circulation. And this is that autoimmune profile. So this is where your antibodies are binding to yourself. And this is typically due to a lack of clearance of apoptotic debris or nets from neutrophils. So these autoantibodies will bind to this excess nucleic acid in circulation. And again, you have macrophages that are dysfunctional, that don't clear that away very well, which only contributes to this increased burden of nucleic acids. So these autoantibodies will bind to this self-antigen, and then that can cause inflammation. And these antibody antigen complexes can then embed in various different tissue. And this is where you see that heterogeneity in how the disease presents, because these complexes can deposit in different tissue. Typically in lupus, we see a lot of kidney involvement, but it can also cause damage all over the body. So we get CNS involvement. Heart disease is very common in lupus. And therefore, again, the diagnosis is tricky. And there's also complement that's used as a measure. So C3 is typically low in lupus because the complement system's consumed a lot more. And then some other inflammatory markers that are typical, but also measured across autoimmunity in general, again, making it difficult to diagnose.
Right. And you have sorry, Jason, this different flavors. Sometimes people say lupus, so erythematosus or lupus nephritis. Is that lupus nephritis just because you see deposition because the kidneys are filtrating these complexes from the blood, or is something specific being recognized that's expressed on the kidneys?
Yeah, that's a good question. So you're right. It's sort of secondary to lupus. So you have this, if the inflammation is in your kidneys, it's typically called lupus nephritis, and it has various different stages of definition depending on how much that damage is. But it's essentially the local inflammation that's going from that systemic blood phenotype and getting into your tissue. And again, there's emerging research now that's getting really impressive, looking at tissue, as is across the whole immunology field. But in lupus, it's very significant because kidney damage is such a significant comorbidity. So looking in the tissue is a really exciting new area of research in lupus.
So you talked about the general antibodies, which are markers of autoimmunity, but also for lupus. Why do you think in general, or is that the main reason it's so hard to get a diagnosis for it?
So I think it's because of that overlap with so many different autoimmune diseases. So the typical panel of inflammatory markers that you would look at can also be found in a whole host of other autoimmune diseases. And because lupus is a disease of flare, so patients will present okay for a while, and then something will trigger their autoimmune response, and they'll get a much more aggressive immune phenotype, which is where you can really see those markers and antibodies come out. So you might not necessarily see them when a patient isn't presenting with a flare, which again makes it tricky to diagnose. So you've got to try and catch those patients during a flare, but you also want to prevent them from having flares using anti-inflammatory therapies. So that's another contributor to why it's so tricky to diagnose as well.
Because I guess in the end, what you need is to have the ultimate confirmation, quote unquote, would be detecting those antibodies in the serum and saying, oh, these are anti-nuclear antibodies, therefore it's lupus. But if I understand, I mean, for those of us that don't have an MD, we might forget that usually to get to that point that people actually get these assays, you probably go over many other less specific blood tests that will just give you not the right answer until you really zero it down? Or are we already testing for these things early on? Jason is shaking his head.
Yeah, I was going to say anti-nuclear antibodies exist in all types of autoimmunity, which is the problem, right, George?
Exactly. and full disclosure as well, I'm coming from a basic science background, so my knowledge as the clinical background is also quite limited. But as I've tried to learn as much as I can over the years of the clinical side of the disease, and the more I learn, the more complex it becomes.
So yeah, getting into your research then, what are you focusing on? I know you use rare patient cohorts as well, but what are you really trying to solve with your research with lupus?
Yeah, so my research is specifically focused on those that develop the disease before the age of 18, which we call childhood onset lupus or juvenile onset lupus. And why those patients are interesting is because the disease is much more aggressive and patients typically have a worse disease course. They have more lupus nephritis. They also have more CNS involvement. And because they're younger, it's important to try and tackle that age because they're kids going through school, a lot of developmental processes, so puberty, which I'm sure we'll talk about later as well in terms of the sex bias of disease. But it's a really important disease category to focus on due to that increased aggression of the disease. So my research has always been focused on these younger patients. So we have an incredible setup here at UCL attached to Great Ormond Street Hospital and University College Hospital, where we were able to get samples through a biobank in the Centre for Adolescent Rheumatology. And we have an amazing resource now of samples from patients that have been followed up for a really long period of time. And that's also improving in terms of tracking them from Great Ormond Street through to University College. And that's allowing us to track disease longitudinally, but also to get a real clinical understanding of the patients. We work very closely between the clinical team and the scientists, so we're able to have that constant stream of communication to really understand the disease and track that basic science research that we're doing back to a clinical setting and really make it translational. And my research has been split into 3 themes, which every time I try and focus on one of the themes, they overlap with each other, as is the nature of lupus. So the first theme is just understanding the immunology of childhood onset lupus. So I've done a lot of phenotyping and understanding disease through multiomics to really address the heterogeneity of the disease, but also look at markers that are common across all patients, which is harder than it sounds. And then the second theme is that cardiovascular disease is the leading cause of mortality for lupus patients. So we do a lot of metabolomics work and understanding how immunometabolism contributes to cardiovascular disease and that increased risk in lupus that's driven by both inflammation but also dyslipidemia. And then finally, understanding the sex bias of disease. So 90% of lupus patients are female. So with that, we want to understand both why the immune system is different in healthy individuals between males and females, but also how that contributes to this increased risk of developing autoimmunity. And what typically happens is that these themes constantly overlap. So you have cardiovascular risk and sex differences there, which seems to break down in lupus, and you have this increased cardiovascular risk in a female predominant disease. And then the immune phenotype obviously contributes to the immunology of both of those aspects. So that's a summary of how that research is split, but I'm happy to go into more detail.
So for the sex bias of disease, why? And do you see it in the juvenile group as well, which would suggest it's something pre-puberty?
Yeah, so it's really interesting. So you do still see it in the juvenile patients. It's slightly less extreme. So it's 4.5 to 1 male to female, female to male ratio in the juvenile onset patients, whereas in the adults, it's 9 to 1 female to male. The reason for that skew is potentially because of puberty. So we see it includes children pre-puberty as well. And then you also have that increased monogenic aspect in the younger patients as well. I think both of those aspects skew that sex bias in the younger patients. But essentially, there's multiple reasons we think why. I think we think it's both sex hormones, but also sex chromosomes. So in terms of sex hormones, we've done a lot of work around defining how they influence immune cell phenotype, both by flow cytometry, but also looking at RNA sequencing to understand what what genes are modified by sex hormones to drive this functional change in immune cells. But we see differences in abundance of immune cells as well between both healthy individuals, but also in lupus patients themselves. What's interesting is that lupus seems to override that sex difference. So whether we look at metabolism or immunology itself, this overarching inflammation seems to normalize this difference between males and females, which is really interesting. And we have sort of indications that the pathogenesis might be different as well. So because you're coming from that different immune cell phenotype in healthy individuals, if you then compare healthy males with lupus males and healthy females with lupus females, you sometimes see different pathogenic mechanisms. So that's also a really interesting aspect to look at. But the overarching thing that we see in lupus is that type 1 interferons seem to be always there, present in the background. And that's where the sex chromosomes come into play. And we know that TLR7 is found on the X chromosome, for example, and that's heavily involved in nucleic acid sensing and the increased type 1 interferon in response to usually viral infection. But in lupus, you're obviously getting these immune complexes bound to nucleic acid that's signaling down that TLR7 pathway, promoting interferon production through plasmacytodendritic cells. And then you're getting that increased inflammation. And then that's going to trigger your autoimmune response against self-antigen. But because TLR7 is on the X chromosome, I think there's an increased risk of that being overexpressed in females. And if you combine that with other lupus triggers and background increased risk factors, that might skew the bias of lupus development towards females. But ultimately, we think it's a multifactorial reason.
So I really like hearing about the TLR7. I think we've known that about that for some time, and it's really the idea that you have some leaking or some expression from both chromosomes. Although, kind of a general understanding is that you have a silencing of a second X chromosome. That doesn't seem to be the case for every single gene in that chromosome. I think that's something that now we're starting to accept that's the case, that the silencing is not perfect. And so I have two questions that maybe you can answer kind of in a package. On the one hand is do we know about specific triggers that might kind of really get the disease started in susceptible people, either the juvenile or the adult versions of lupus, and whether these which exactly are the genetic predispositions that you might see, and whether those are more represented in young patients, and how do you kind of put that together?
Yeah. So in terms of the genetic predisposition, we often see with the research in that field is really booming at the moment. So increased technologies around genomics and the sequencing is helping us to understand the disease a lot better in terms of both the polygenic and monogenic form. And I have a lot of colleagues and collaborators that are really leading in the field for that. And that lends itself a lot more to the childhood onset and juvenile onset patients because of that increased monogenic format and understanding which specific genes might be contributing to disease in those patients. But that ultimately helps us to understand the disease and other diseases in general by looking at those genes in general. They are very often involved in interferon signaling, but also they're involved in T-cell B-cell interaction and lots of other innate and immune responses. Also in the complement system, so you see mutations in complement proteins. And yeah, so that genetic factor is definitely there. And we're understanding that a lot more. And I think it is a combination between having that risk combined with other factors that increases that risk. But when you combine that with triggers like UV radiation, viral infection, but also stress and diet and things like that. So it's that creates a perfect storm in these patients that are already predisposed to autoimmunity.
So then in terms of therapy, you'd mentioned CAR T cells. Are they acting on this in some way?
Yeah, so the CAR T cell therapies is a new way to target B cells. So we're trying to deplete B cells in lupus to stop that autoantibody production. Typically, monoclonal antibodies like rituximab we use to target B cells and deplete them, but they don't necessarily get to those tissue resident B cells. So you sometimes still have some remaining B cells with that autoimmune phenotype in tissue, and they eventually they would reemerge and kickstart that autoimmune phenotype again. But with CAR T cells, they can really wipe out and completely refresh that B cell phenotype. So when they return, they don't necessarily have that autoimmune profile. And so far, patients that have been on this treatment have largely been in remission, and it is looking very promising as a therapy. It will be the long-term tracking of those patients that will be the true answer, but so far it's looking very promising. This obviously comes at a cost and a burden on the hospitals, that type of treatment. So we'll still need a lot of development around. There's a lot of anti-interferon therapies that are proving to be successful in lupus, and they're currently being trialed as well. Some of which are approved. So anifrolumab is a monoclonal antibody that's already approved, at least in adult patients for use, and those pediatric trials that are ongoing now. So hopefully that can be a promising new treatment for the younger patients. So you quite often see that lag between adult trials and pediatric trials, which tends to delay the treatments for the younger patients. But hopefully that things like that will improve in the future.
So for some inflammatory disease or some autoimmune diseases I don't know if this is okay for SLE, but there's some diseases for really young children that they kind of grow out of them, or that they kind of tend to resolve themselves as the child kind of grows up. Is that the case? Do those any of these juvenile patients just remit on their own? Is it always a kind of a growing progression if the people are not treated, or what is the success rate of early interventions for these patients?
Yeah, again, it's a good question, and I guess this is where my lack of clinical background will present itself. But patients can go into long-term remission, sometimes in response to treatment, sometimes just as a result of that inflammation going down and the triggers not being as present. Because it's so multifactorial, sometimes if you just take away one of those risk factors, it could improve. I think even diet has sometimes improved symptoms for patients. We've done some work engaging with patients about how diet's improved their self-reported symptoms. And some patients really swear by certain diets and dietary changes that have helped their lupus. So yeah, in answer to that, I think it's complex, but patients can go into that kind of remission themselves with the right treatment, and trying to get more of those patients. And I don't know the numbers on that, but getting more to have long-term healthy lifestyles would be amazing.
Yeah. And one more thing I also thought it was very interesting that you were studying the effects of gender affirmation therapy in young patients. And this reminds me of another colleague. We had Peter Broding with us also many episodes ago, also discussing studying this, because it's a really nice model in which you can put apart the effects of the genetics and the effects of the hormones, and that gives you a unique study model. So what have you learned from these studies? Maybe something that was surprising for you when you were analyzing these patients.
Yeah, Pettis did some amazing work, and we're also using this opportunity to look at that, I guess, in vivo opportunity to look at how sex hormones influence in the immune system, both dependent and independent from chromosomal background. So in transgender individuals, we're able to see how gender-affirming sex hormone therapy can both modify the immune system, but also metabolic profiles. So we're doing work around immune phenotyping, but also how the sex hormones influence systemic metabolism. And I think personally, that's been the biggest surprise, is the effects that they have on metabolism. So we see a big, which kind of lends itself to that cardiovascular aspect that's associated with sex. So we see with a background of estradiol, you get this increase in atheroprotective HDL. But when you lose that protective effect of estrogen and have testosterone in circulation, you lose the protective HDL profile, but develop more of this atherogenic VLDL profile. But again, in lupus, when we compare those sex differences, they're gone. So something about the autoimmunity sort of levels that cardiovascular risk, I guess. So that's been quite a surprising finding. But as we delve more into the immune system, we're finding big effects on specifically B cells we've looked at so far. So work with Kosiana Chet and Lizzie Rosser, Hannah Peckham. So we've found some really interesting differences in B cells. And I've been working on Tregs myself, looking at how sex differences and sex hormones influence Treg function and number. So they've been really interesting studies, and we're delving deeper into that now using spectral flow cytometry to look at a bigger picture, kind of zooming out again, increasing our cohort numbers and trying to find specifically how those chromosomes and hormones influence disease. We also have cohorts with Turner's and Klinefelter syndrome, so we're able to tease out how the chromosomal background and dose also influences the immune system. So that's going to be really interesting going forward.
Interesting. Well, so it sounds like we're going to have to keep an eye out for a few papers coming forward. So that being said, we always try to end the podcast with kind of a adjacent question to the science, as it were. And so for you, what's the best piece of advice you've ever received, professionally or personally?
So it's a bit of a funny one, really, for the best piece of advice is that no one has a clue what they're doing. Because I think if you have that mindset every day, I think it helps you to relax a little bit and be a bit more silly with your work. And and I think that's true if you ask anyone if they know exactly what they're doing. I don't think anyone does, really.
It's like that advice that if you're nervous giving a talk, or or an actor is nervous, it's like they say, just imagine that everyone else is naked, so everybody is even a worse situation than you. So yeah, I like that advice. Do you know what you're doing, Jason? Would you say?
Only rarely.
Only rarely. All right. So that was super interesting. And I personally am so excited about the whole CAR T-cell therapy advances for autoimmunity. And it's like, this just scratched the surface. There's so many indications that this could work for. So I was super excited when I saw the first papers about this. So I'm really looking forward to see, all these people that have these terrible, terrible diseases can be offered a viable alternative. So we'll see what the future brings to this. And so we had Dr. George Robinson from the University College London today at the podcast. Thank you so much for joining.
No, thank you very much.
Thank you for coming on.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast, Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback, or if you have a suggestion about a guest. See you next time.